right. Well, thank you everyone for joining us today for another Diatom Web Academy. The Diatom Web Academy is brought to you by the Taxonomic Certification Committee of the Society for Freshwater Science and diatoms.org. You can keep track of upcoming Diatom Web Academies at the diatoms.org news link. To see what's coming up, we'll announce what the next couple that we're going to have at the end of today. We're in season 2 of Diatom Web Academy now. We've been doing this for a little over a year, trying to bring everyone together for every couple weeks to form a little bit of a community from around the world, around North America, to discuss diatoms, discuss assessment, discuss monitoring, and discuss how diatoms are used. All of our webinars are recorded. In a couple days, you can find even today's chat on our YouTube channel. Again, you can find those links at diatoms.org. How we'll handle today is in just a second, we'll introduce today's speakers with Sylvia. And if you have questions, please pose them in the chat. And at the end of the presentation, Sarah Spalding will handle the Q&A and moderate that and leading up towards the end of our hour. So thanks again for joining us today, everyone. And I'm going to turn it over to Sylvia Lee, who's going to introduce today's Diatom Web Academy speakers.
Thanks, Mark. It's my pleasure, great pleasure, to introduce today's Diatom Web Academy speakers. Dr. Evelyn Gaiser is the endowed George Barley Eminent Scholars Chair and Professor at Florida International University in the Department of Biological Sciences and Institute of the Environment. She is the lead principal investigator of the Florida Coastal Everglades Long-Term Ecological Research Program. And she is not only a brilliant scientist, but also a wonderful mentor to students, including myself. And she is a trained musician, creatively combining science and music. Another creative and talented person I'm fortunate to know is Franco Tobias. He has been an integral part of the Gaiser Periphyton Lab for many years. As lab manager, he conducts field work, trains students and interns, and other behind-the-scenes work that keeps the lab running smoothly, and is just so important for the morale and productivity of all the lab members. And he does that all the while taking care of his beautiful twin daughters. And with that, thank you to the speakers, and I'll hand it over to you, Evelyn.
Wonderful. Well, thank you so much, Sylvia, for that such nice introduction. It's great to see you and everyone here. And I just wanted to thank you and the team for running this series over the pandemic. It has been so important for everyone in our lab. We either come or we listen to them on YouTube afterward, and we've learned so much, and we have felt connected to all of you. And we just thank you so much, and really glad to be a part of it today. So as Sylvia mentioned, we have been conducting these studies of calcareous benthic diatom assemblages in the Everglades for many years. I started at FIU in 1997, and Franco has been working on the Everglades diatoms and organizing our field collections and lab protocols for almost all of those years. So it's fun to be able to give this talk with him. These communities are pretty esoteric in the Everglades, so I'm going to spend some time just introducing you to the incredible periphyton mats of the Florida Everglades. I think this talk will be of interest to folks who are interested in wetland benthic algae, particularly these weird communities that we tend to find in subtropical and tropical wetlands that have been less well studied. As well as folks who are doing assessments, and we have been developing tools for hydrologic and water quality assessments that are working really well based on the Everglades diatom and flora and other features of the periphyton that we'll talk about. So we're calling out this, back to the first slide there. We're calling out this beautiful diatom here. It's our most famous diatom, named by our most famous diatomist. It's Mastigloia calcarea Sylvia Lee. And we'll talk more about this diatom in a little bit. Okay, next slide. Here's a little outline. I'll give you a little Periphyton 101, talk about monitoring and assessment protocols that we use. And then Franco is really gonna dig into our Periphyton and diatom database. And then we will come back and I'll come back and talk a bit about what we're learning about long-term change in the system and how we might apply what we're learning to other subtropical and tropical wetlands. Next slide. So first, the Periphyton of the Everglades 101. Next slide. Okay, so Periphyton is really a predominant feature of Everglades wetlands. You cannot miss it. It is everywhere and it's really abundant. This map on the left is of our Florida Coastal Everglades LTER domain of Everglades National Park. And it shows where we find freshwater marsh, mangrove, and seagrass habitats. And in all of these, we find copious calcareous benthic mats growing either on the surface, draping over plants and over the bottom of the limestone bedrock, underneath the mangroves and attaching to their prop roots and even over the seagrass meadows of the Florida Bay estuary. Next slide. And what we find about these communities is that they're really well adapted to the extremely low phosphorus conditions that exist out in the middle of the natural Everglades. Typically the Periphyton the water total phosphorus concentrations out there range from maybe 5 to 7 parts per billion. And that is ultra oligotrophic conditions. We're just a phosphorus starved ecosystem. They're also really well adapted to seasonal drying that is typical of subtropical habitats. You know, we have this distinct dry season that we're coming into right now and a very prominent wet season in the summertime. And so the areas that are shallow dry out for a large portion of the year. We call those short hydroperiod marshes, in contrast to wetlands that dry only for a few months or maybe every other year or so. And those are called the long hydroperiod marshes. And they have slightly different types of periphyton that we'll talk a little bit about. Next slide. Another weird feature of these mats, a very distinctive feature, is that because we are in this limestone-based environment, the mats precipitate out calcium carbonate. And primarily this is done by the filamentous blue-green algae. Our two dominant species are Schizothrix calcicola and Cytonema hoffmanii, and they form kind of the backbone of the mats, and they're precipitating out these calcium carbonate crystals and forming a home for all the other algae, including coccoid blue-green algae and green algae and diatoms, many of which secrete this extracellular polymeric substances, or EPS. And we see this beautiful Mastigloia calcarea exuding EPS from the little pores along where the protecti are along its girdle. So next slide, that's great. What we're finding about these communities is that they are really ecosystem engineers out in the Everglades. They regulate water chemistry, the concentrations of nutrients and gases. They precipitate out this calcium carbonate, and that forms our marl soils. They're full of fungi, bacteria, cyanobacteria, algae, and so they contribute substantially to biodiversity. They're at the base of the food web, although their food quality is not that great because of all the carbonates in there. They are important homes for a diversity of aquatic consumers. And what we're going to talk mostly about is how they respond really quickly and distinctively to changes in hydrology and chemistry. Those are the primary drivers that are changing in the Everglades ecosystem. Next slide. And this ecosystem has changed substantially in the last century. On the left is this recreated satellite image that shows how the Everglades used to look with sawgrass plains and ridges and sloughs and sheet water flow all the way from Lake Okeechobee down to the Gulf of Mexico. And then we drained all that. We put in thousands of miles of canals, sending that fresh water to the sea in order to develop the land right under Lake Okeechobee at the head of the Everglades watershed for agriculture and expand all this urban area for development. And that resulted in a smaller Everglades, a dehydrated Everglades, and a polluted Everglades, polluted primarily with phosphorus that's coming from all this agricultural area. So this led to the federal government suing the state of Florida. The Everglades Forever Act that called for establishment of a nutrient criterion to protect these wetlands from further phosphorus enrichment. Next slide. That was something that drew me to South Florida. The first project that I participated in was establishing a protective criterion using an experiment that is very similar to one that Jan Stevenson talked a lot about, that was used to set this water quality protective standard for phosphorus. And this is a flow through flume experiment where we added 5, 15, and 30 parts per billion of phosphorus, let that flow downstream with the flowing water in 3 replicate systems in Everglades National Park. And very quickly, we saw a lot of changes occurring in the periphyton community that we followed for 5 years. And shown here is this on the, well, we see we're looking down on a meter square quadrat in the control channel there that shows a lot of periphyton, and then the 30 part per billion channel showing the periphyton just disappears. And that's what we find distinctively occurring in the Everglades in response to phosphorus. And on the right is this table that shows the different response parameters throughout the ecosystem and how long it takes each one of those to respond. And what we see is that periphyton phosphorus content, other microbial indicators in the flock, diatom species composition, periphyton cover are all early responders and are really triggers of this ecosystem response cascade that occurs through time, that occurs at all levels of exposure. Next slide. And so we were able to establish these periphyton quality, quantity, and composition metrics of response to phosphorus enrichment. And this work helped establish the class 3 water nutrient criterion set at 10 parts per billion for inflowing water to these protected areas. Next slide. So the next frontier really of Everglades restoration, now that we have this protective water quality criterion, is reestablishing freshwater flows into the system and doing that as sea level is rising. So in the map again on the left is the smaller arrow indicating the freshwater flows that are so depleted at the same time that we're experiencing a really accelerating rate of sea level rise. And that causes 2 things to happen. We have a desiccating ecosystem that's losing carbon from the periphyton to the soils. And next slide, we're also, accelerating saltwater intrusion into the interior even faster as a result of this combination of sea level rise and depleted freshwater flows. So we've been studying this, and now the next slide shows some summary of the work that Sylvia did for her dissertation. This is a figure from one of her papers related diatom composition to hydroperiod, or the length of time that the marsh is flooded with water. And she found a really strong relationship there and came up with some key hydrologic indicator diatoms that Franco is going to revisit when we get to his part of the talk. Next slide. And then Viviana Mazzi came in the last, she finished up her work a few years ago, summarizing the effects of saltwater intrusion on these communities through both experimental work and surveys along salinity gradients that allowed her to determine what are the indicators of salinity along these gradients. And she did some explorations with the Titan method for understanding what those salinity or conductivity thresholds are for fresh and brackish water diatom communities. And Franco will show you some of those indicators as well. Next slide. So it's really critical that we are finally at the brink of whole ecosystem scale freshwater restoration of this Everglades ecosystem. We've had an ecosystem that's enriching, one that is drying, one that is getting saltier. And so restoring clean freshwater flows is imperative. We've known this for decades. We're finally seeing it happen. This is a commonly used map to show, a figure used to show in the middle, current flows all diverted to the sea, the historic flows on the left with the sheet flow that I showed you before, and then on the right, where we hope to go with Everglades restoration. And in the little circles, I'm just calling out 2 projects that are underway that we're already measuring successful effects of, which is super exciting. Next slide. These 2 projects include the bridging of the Tamiami Trail, a major blockage to flow into Everglades National Park. And you see the new bridge there that was installed in 2013 and another one more recently in 2018. And then filling of canals along the eastern boundary that's allowing the system to retain more fresh water and send more fresh water all the way down to Florida Bay. Next slide. So now what our lab is really focusing on is monitoring and assessing ecosystem response to those restoration projects. Next slide, I'll talk about how we do that. We do that in concert with measures of lots of other indicators from periphyton up to crocodiles. And we established this methodology and we chose these indicators based on their array across these temporal and spatial axes of restoration. And we see periphyton down there at the bottom of this figure showing that it's able to indicate restoration across the whole spatial domain of the Everglades because it's everywhere and it responds really fast to change. And on the right here is the map that of all of our sites that we visit every year to assess how the Everglades is doing through, we'll use this abbreviation, SERB map. It's our assessment program where we measure how the periphyton is doing relative to all these projects going on to restore fresh water. Next slide. This program assesses 2 hypotheses, one about trophic status. And for that, we're really assessing edibility, how palatable and energetic the periphyton, how efficiently the periphyton energy can move through the food web. And we do that again with 3 metrics of quality, quantity, and composition. And next, the nutrient hypothesis that again tells us about departure from oligotrophic conditions based on, again, 3 metrics in these same categories: the periphyton P content, its biomass, and its oligotrophic diatom composition. Next. And our assessment protocol follows some pretty commonly used approaches where we consider all of these points in the ecosystem to contain both reference conditions and impact conditions. We look at the frequency distribution of each of these attributes across all those sites. And we consider the 75th percentile of those conditions to represent baseline conditions, and a standard deviation away from those baseline conditions. We tag those sites as altered, and we set those expectations in a basin-wide method using criteria that are specific to each one of the management units of the Everglades for each one of the multi-metric criteria. Next slide. A little bit of work that we did to understand the importance of including diatoms in a multi-metric. And we were able to show using our long-term data from our flume study that the probability of correct detection using the methodology that I just showed you increases with our incorporation of diatoms into the multimetric by about 15 to 30%, depending on whether we're looking at low, medium, or high level of exposure of phosphorus to these communities. So the diatoms are a really important, no surprise to any of us, component of these multimetrics. We've shown that in many different ways. And now I'm going to turn it over to Franco, who's going to talk about the beautiful diatoms that we have in the system and how we measure them. Franco, I think you're still muted. Okay, there you are. Oh wait, you're still there.
There. Thank you, Ev. Good afternoon, everyone. My name is Franco Tobias. For those of you who don't know me, I am Evelyn's lab manager, and I have been a member of the Paraffine Analysis Lab for the last, well, I would say this would be my 22nd year. And I was hired way back in 1999 out of the University of Alberta. And my first experience with diatoms came through identifying flora from Alberta's lakes and ponds. And I'm here to dive a little bit into paraffine assessment and the development of our diatom database. Now, I can consider myself lucky that part of my work has given me plenty of opportunity to explore South Florida, especially given that the span of our work covers a good portion of the bottom half of the state. It's wide-ranging from a spatial perspective, with over 3,900 unique coordinates sampled so far. The temporal component is also extensive, with 20+ years of sampling overall, and some projects spanning over 10 years and counting of continuous data collection. Our diatom research covering this large area in space and time has really provided great insight into ecosystem function and the effects of restoration. Now, when I was first hired, the lab was really involved in monitoring projects that assessed the impact of phosphorus enrichment, as Evelyn said, on diatom communities in the short and long term. She mentioned the Flume project, so I won't elaborate much more on that. But at the same time, we were also involved in the EPA's remap, which traced the effects of phosphorus inputs on different portions of the Everglades ecosystem, covering areas from Loxahatchee to Everglades National Park. Now, in continuing with the lab's work in the early years, we also started looking at diatom community responses to hydrologic change. And it was around this time that the FCE LTR program really began to develop and expand, using paraffine, among other ecosystem components, to look at the causes and consequences of long-term change on coastal ecosystems. Hydrology and its management also had an effect on endangered species, and we saw that by being involved in some monitoring projects related to the Cape Sable seaside sparrow, where we used paraffine as a metric to determine how water management affected the bird's habitat. As you can see, one of the insets here where Evelyn is in front of the helicopter, she was doing a paraffine assessment and collection just to determine whether the presence and absence of paraffine had an effect on the type of macrophytes that the birds would use for nesting. Now, all of this work was happening in one of the most unique ecosystems in the world. And one of the challenges we face is, is how to get around. As you can see here, we have our ways. I would say PD Eastman's Go Dog Go sums it up quite well. We can go by boat, by car, by helicopter, or by foot, as you can see here. Now, of course, since our choice of study is paraffine and diatoms, we have to go where they are. But that's not always the case. As Evelyn said, we do have mats of paraffine, massive mats, but they may be absent. And so where our work is to follow the paraffine, we have to go to these places as well and ask the question, so why are they not there? And so we've had the pleasure of being able to sample what we can consider a landscape mosaic. We have Loxahatchee National Wildlife Refuge, LOX or WCA 1A, water conservation areas 2 and 3, just south of that, and then Everglades National Park divided into Shark River and Taylor Sloughs. It's a massive area. We're talking about 17,000 square kilometers, and with habitats ranging from marshes with tree islands to the north, as you can see, that's kind of the center top photo, to rich and slough landscapes with cattail and sawgrass dominating, and then to open sloughs with spike rush and water lilies, to short hydroperiod marshes with pinnacle rock heading south to the coastal areas dominated by mangroves. Now, when I mentioned these monitoring projects, they really allowed us to have a greater understanding of the diatom communities that exist in these different habitats. We know that they have distinct spatial patterns, and we gained that through all of our monitoring sampling. And they are defined by nutrient availability, hydrology, pH, and conductivity gradients. And so seeing these diatoms and learning how they respond to environmental change really gave us the opportunity to dive deeper into our Everglades flora. Now, our initial flora, or initial voucher, which we made available online in its early stages, was the beginning of our cataloging diatom diversity here in South Florida. In its earliest form, we were guided by Podzorski's work in the Caribbean, of course, Patrick and Reimer, Kramer and Longoberlo, and other European authorities. And we used MOTUs for the taxa we couldn't quite fit into the ones in the literature, with the goal, of course, being consistency and identification. And our monitoring work really expanded our use of diatom composition as purified metrics because of the responses to nutrient and hydrologic change. And that made developing our taxonomy even more crucial. And Jennifer Slate and Jan Stevenson's 2007 work really became instrumental in doing this. And our collection of images of diatom flora from a wide range of habitats allowed us to expand their initial findings. And you can see some examples here. In terms of indicators for hydroperiod, we were finding that Fragilera grotesca was very good at indicating shorter hydroperiod areas. And then Ensynema mesianum becoming more dominant in some of our longer hydroperiod areas. In terms of the oligotrophic system, Caponia caribia, which is fairly unique to the Caribbean region, as the name implies. We have Brachycera microcephala, Diploneus oblongella, Ensynema evergladianum, of course, named for being down here, Sylvia's Mastagloia calcarea, and Nitrus serpentarate. And in contrast, we have our eutrophic diatoms, the Rhopalodia gibba, Nitya amphibias, and Gomphonium parvulum. Now, our collection of these images helped us learn some more about these species. And so we were able to also provide some nomenclatural updates as new descriptions became available based from our studies. Now, along with nutrient and hydrologic change, our diatom work also helped generate assessment metrics for purifying quality in relation to biomass, what Evelyn was talking about in terms of edibility. And so we have some diatoms that really like it when there's not as much calcareous material, that being, Ensynema evergladianum being one of them. And then it's just serpentarate, which really likes the very calcareous mats that we have present in these areas. The diatoms are also good for defining areas affected by a combination of nutrient availability and salinity. And so we have our brackish eutrophic species, such as Fragilera Mastagloia brownii, salinicola, and Epithemia pacifica, to our oligotrophic stuff, which is Mastagloia lanceolata and tenera. Into the freshwater, where we have, again, some of the species you see here, the Gomphoniums that I named earlier, and the smaller diatoms like Ensenopsis microcephala and Achinanthidium minutissimum, which really like oligotrophic stuff. Now, the salinity issue is really becoming a greater concern to coastal areas, and so having these indicators are of great importance. And so we see in the early stages, our lab's monitoring work really paved the way for developing purifying-based metrics for environmental assessment. However, we knew that moving forward, we really needed to come up with standards for sample collection and processing that will enable us to gather data that we can integrate from all related projects, and that takes into account the differences between sites, samples, and seasons. And so to this end, we came up with what we call our project workflow that condenses our field collection and sample processing into a schematic that outlines all the bases for SOPs. It generates sampling efficiency and data coherence and makes it a great training tool for new personnel. And that's because consistency in dealing with samples became important once we became involved in assessment projects, especially with the biggest project that Evelyn was talking about, the implementation of modifications to water flow as a pathway to Everglades restoration. Now, we saw the development of projects that measured the effectiveness of this restoration plan at the regional level, These include projects, what Evelyn mentioned before, Northeast Shark Slough, the Saline Everglades near Biscayne Bay, and the eastern boundary work in Everglades National Park, and adaptive management strategies in Upper Taylor Slough. These are at the regional level under the overarching large-scale projects conducted within SERB. Now, how large is SERB? Now, we're talking about, it was implemented in 2004 and continues today. It consists of about 150 sites randomly distributed throughout South Florida. It's visited every year. These sites are visited every year, depending on water availability and sampling access, which means that since its inception, we have sampled more than 2,200 individual coordinates. Now, it's a multi-level assessment on a large scale and generates a lot of data. And if you can recall, it covers a large swath of land, and the most efficient way of transport is by helicopter. Now, there are advantages to this. It's quick from point A to point B, relatively minimal impact on the environment compared to an airboat. The only downside is cost. And so this is where efficiency really is important. The regions that I mentioned before are divided into randomly dispersed primary sampling units, which are then divided into 50 randomly generated draws, from which one is chosen for sampling by a throw trap with 3 throws as replicates. Now it's, I think if I, in layman's terms, I would say it's like taking a snapshot of the sampled area at that particular time. And so what kind of information do we get? Well, what we can see on the side here, there's kind of an array of photos. What we see within a throw will vary with water depth, with plant composition, whether there is paraffin or not, and just the general habitat of the area. But we collect physical parameters from the water, measure paraffin biomass, coverage, and type. We record the associated substrates, we do vegetation composition, and eventually we collect paraffin and consumers. Now, the scale of this project has generated so much information. We're talking in the tens of thousands of analytical data points that is best summarized by our paraffin database. Now, I won't dive into the details of the database mechanism, but it's a means of organizing all the information we collect from SERB into the 4 paraffin-based metrics we use for assessment. These metrics, in turn, answer questions generated from monitoring work to assess the effectiveness of modified water deliveries on restoration. Evelyn will elaborate more on this point in a little bit, but let me highlight the diatom work that we have done in our metric for composition. What started out as our voucher cataloging diatom diversity really exploded with SERB and has developed through the years into the Paraffin Analysis Lab's diatom database. Now, we had our initial voucher flora, which underwent major taxonomic synchronization. We prepared them for use in future assessment work, which included SERB. Sylvia, as a student in our lab, continued work on synchronizing names with further research updating our database. Nick Schulte, who was a student in our lab, also in turn designated metadata that helped us identifying distinct populations from measuring and photo documenting a range of representative specimens for each taxon. And now our current iteration is undergoing growth through continued updates in names as well as additional SERB counts. And so our database currently contains roughly 420 taxa, 2,700 photos from 161 species with about 12,000 measurements, 120 descriptions so far with 13 autocology descriptions. And our goal really is to consolidate the data we gather from SERB and all of our other projects and come up with a taxonomic database that not only identifies our key players, but also incorporates current nomenclature, photos, measurements, relative abundance maps, and environmental information. And we also hope to include data generated from our paraffin metrics to provide greater resolution, especially with our indicator species. And so our next slide is kind of a sample outline of what we are thinking of including for our future publications, although I'm sure this will be subject to modification throughout its development, with our ultimate goal of furthering the understanding of South Florida ecosystem through its diatoms. Now, of course, this can't be a diatom talk without meeting the main characters. And so here's a representation of what we have been able to photo document so far, with some of our indicator species identified by the different colored circles. And as you can see here, some species are used as indicators really for more than one condition. Now, while working on this diatom community record, we were also able to expand on historic taxonomic work on Everglades species. When we first started, we began characterizing the morphology and ecology of the Everglades Comphanema complex, which is an important indicator of eutrophication in the system. And then Sylvia, along with Evelyn, Bart Van de Veer, Mark Edlin, and Sarah Spalding, worked on an important Everglades indicator species of oligotrophic, eventually describing two new species, what we have on the right, Mastogloia calcarea and Mastogloia pseudosmithii. Sylvia's work on synchronizing our taxa also resulted in the description of a new species, Envicadia metzelsinii, from karstic wetlands in Everglades, as well as contributing to works on the transfer of Stauronace pachycephala into the genus Envicadia. And Viviana Mazet, while working on her salinity project, also worked on Ensynema evergladianum and its ties to microbial biomass within the Caribbean region. And so these are works that we have done so far. We have done some other taxonomic work on areas and species not included within SERB, such as our amphoras and seminavis. And as always with taxonomy, we find what we would say problematic species that will require more documentation and research and names. Now, developing our diatom database also provides an opportunity for our students to conduct their own research into specific Everglades core taxa and eventually make contributions to DONA. And so Sylvia, Nick, Vivi, and Meredith Emery, one of our current students, have submitted some taxa to DONA, and we look forward to adding more Everglades diatoms to this list with time and effort. And maybe with some help from Katie Johnson, who's currently doing some of our soft algae work. And now kind of to end my portion of the talk, and as a final thought before I pass the baton back to Evelyn. We know that understanding the drivers of ecosystem change is more complex really than can be told by any one group of organism. And diatoms as indicators cannot tell the story alone per se. But using them within paraffin-based metrics of assessment have really played a big role in developing the story of Everglades ecosystem restoration. And so I'm going to pass the talk back to Evelyn as she talks about long-term dynamics and cross-system applications.
Thanks so much, Franco. And those animated slide, well, the one that you just showed of the changing abundances of our two core diatoms and the two important drivers of those changes. It's just a snapshot into what we can learn about variability across the system and over time. I want to dig a little bit more just into how we've been applying our stoplight tool that I talked about. And then just briefly about how we might apply what we're learning about the Everglades to other subtropical and tropical ecosystems. Next slide. So this is our 2019 multimetric periphyton stoplight. This is one component of the multi-indicator report with all those other organisms that I showed you. This gets delivered to an entity called the South Florida Ecosystem Restoration Task Force, and they deliver it straight to the US Congress. Congress is in charge of overseeing Everglades restoration, so we have to have this very simple way of saying, how is it going? And so we code each one of our map sites with this really simple green, yellow, red stoplight, and we can see changes in the quality of the system over time. Some of these areas in water conservation area 3 that show up red continue to receive too much water that's not meeting the water quality standard. Interestingly, we have some red sites appearing only in recent years along the coastal margins, and those are sites that are receiving additional phosphorus as a result of saltwater intrusion that's bringing marine supplies of phosphorus into the interior. Next slide. We learn even more when we zoom into these regional scale studies that Franco mentioned. So we use the SERP map for this large scale assessment. But for each one of these regional projects, we can apply our stoplight tool as well. And I'm just zooming in here to our Eastern Boundary project that's documenting the effects of, you see here, a crane putting backfill into a canal to stop up the water there and to rehydrate the interior of the Eastern Everglades. While that's going on, we have legacy nutrients that had built up in the system prior to having that protective nutrient criterion. And these rehydration projects are actually mobilizing some of that legacy phosphorus into the ecosystem. And the main way that we're documenting that transport of legacy nutrient and encouragingly, the depletion over time of the legacy nutrient due to sequestration by these periphyton communities is via this periphyton stoplight tool. We work really closely with our Everglades National Park managers to assess the quality of the system from the eyes of the periphyton and its diatom components. And they react to our annual reports by actually adaptively managing the way that water flows into this ecosystem. It's pretty exciting stuff right now as we see full-scale implementation of restoration occurring in the Southern Everglades. Next slide. So addressing this question of how can we take the powerful tools that we've developed for the Everglades ecosystem and use them elsewhere. We've tried this out in other regions of the Caribbean basin that include these kinds of, that harbor these kinds of limestone-based wetlands. We've been able to visit the New River Lagoon in Belize, shown here. Next. The Siankaan, beautiful biosphere preserve that looks for all the world exactly like the Everglades in the Yucatan Peninsula of Mexico. Next slide. The Black River Morass, Jamaica that Josette LaHaye took us into, also karstic wetland with abundant periphyton mats. And next slide. We were able to show through a big meta-analysis how these karstic wetlands and other places compared to the Everglades and to other aquatic benthic ecosystems. And so in this figure, the top figure is showing periphyton biomass, and the bottom figure, the phosphorus content of periphyton in everything from wastewater effluent to freshwater marshes, ponds, rivers, salt marshes, streams. And then we have our Everglades and the karstic wetlands there. And what we see is this extraordinary biomass in the karstic wetlands and in the Everglades, despite extremely low phosphorus content compared to these other ecosystems. Next. And so Josette LaHaye really dug into this and particularly was interested in the comparability of the diatom flora across these different karstic ecosystems. And what she found was that some of the species that we had been used to seeing only in the Everglades that had never been seen elsewhere were found all over these other karstic systems in the Caribbean. Interestingly, some of the more lower latitude subtropical systems and the tropical ecosystems had greater species richness than the Everglades, but many of the same species and much the same response of this flora to phosphorus availability, and suggesting that we can apply the same kind of stoplight tool that we've developed for the Everglades in these other karstic wetlands. Next. Luca Marazzi was a postdoc recently in our lab, and he brought another comparative opportunity to us. He did his dissertation work in the Okavango Delta and explored the periphyton and diatom communities there. And the Okavango is not a karstic wetland, but it is in the subtropics. Interestingly, it had also much higher species richness than the Everglades communities. It also provided a means for testing some theory about functional trait distribution across these subtropical habitats. And we found that the Everglades and the Okavango periphyton communities share functional response traits to these primary drivers of desiccation in the subtropical wet and dry season and extreme phosphorus limitation that also characterizes the Okavango. Next slide. So we are concluding from this comparative work in the tropics that it may be possible to apply some of the well-developed tools that we've spent decades developing in the Everglades to conserving these extremely fragile habitats in the subtropics and tropics that are very quickly experiencing the same kinds of dramatic changes that the Everglades began experiencing over 100 years ago. And so we're working with these local communities to talk about ways in which we can prevent the kinds of changes that we've seen in the Everglades from occurring, and using periphyton as a tool there for monitoring and assessment. Next slide to wrap up. So just to summarize, we've shown you that the periphyton in the Everglades is highly distinctive and weird and unusual in many different ways. It has very high biomass. Its phosphorus content and diatom communities are highly diagnostic of hydrologic and nutrient variability and can be used to assess the effectiveness of Everglades restoration. We do have now a pretty substantial and well-documented periphyton database, and we're employing it to track ecosystem-wide changes and report on those to guide adaptive management. And we think that the system-wide and regional assessment development that we've done here can offer guidance for adaptive management, both in the Everglades, where we are actively doing that, and in other threatened subtropical and tropical wetlands. Next slide. And I just want to call out some of our funding agencies that have supported this work over the years, and thank very much all of the members of our periphyton lab over all the years that generated the data that Franco and I showed you today. And next slide is just a call out to our next Diatom webinar that is going to be given by Xavier Cartata, who's an artist that we've been collaborating with for many years. And I hope you all can attend the next one, because it's going to be all about diatom, glorious diatom art. And we'll be happy to take any questions that you might have. Thanks so much for attending and for your time.
Thank you, Evelyn and Franco, for excellent talk. This is just really exciting what you guys have presented here. And thank you to your students and everyone that you've worked with, because this work that you've done over these decades has really, I mean, you've really covered all the aspects of being able to take science to policy, applying your discovery. So this is just a wonderful talk. Thank you so much. And there's some questions coming in, so I will hold mine and get to the questions from the audience. Peter asks about the Everglades species in low phosphorus that are specific to low phosphorus. Are they found in faraway places like Australia, South Africa?
Yeah, that's a great question. And we have, these communities are fairly distinctive of Caribbean karstic wetlands. That's what our cross-system comparative studies, in the Caribbean have shown us. We do have, we have collected some in Australia and the serendipitous visits there and also in New Zealand, and from carbonate-based wetlands there. And really interestingly, there are no species level commonalities, but a lot of the same genera and same higher groupings of diatoms doing really similar things, producing mucilage strands and hanging out in these similar looking mats in similar ways. But it is a very different flora in the southern hemisphere and sort of Pacific areas that have been, whose karstic wetlands have been studied. That's a great question though, that needs more work.
Okay, Mark asks, he says, you mentioned one measure of periphyton composition as percent edible biovolume. What criteria do you use to determine what's edible and what's not?
It's a weird word, edibility. But we found in the Everglades that it's mainly driven by how carbonate, how much calcium carbonate is in this community. And there are certain species that are pumping out calcium carbonate crystals, and it's mainly the Cytonema huffmanii and Schizothrix calcicola are really abundant. Filamentous blue-greens, they're long and stringy, they're full of carbonates, and we are making assumption that for those two reasons, and also because a lot of blue-green algae produce toxic chemicals, that they would be less palatable for the consuming organisms living in the mat. We have done quite a bit of work to test that idea. What the metric is actually the ratio of diatoms, coccoid blue-green algae, and green algae to these filamentous cyanobacteria. It's a really simple metric that we get from our soft count data, biovolume data. And we've found in some path analyses that we've done using those the great big 150 site data set, that it explains a huge proportion, I forget the percent, but it's a really meaningful proportion of variation in the biomass of consuming organisms in the mat, that really simple ratio. And so we're following that up now with some fatty acid analysis to see whether there's some energetic sort of signals that we can get from these communities to make a little bit more sense of that.
And Jan asks about your response from public stakeholders. So have you had much interaction with those stakeholders, and who are they that are interested in your results, and what have they been most interested in?
I would say that our we're in constant interaction with the Army Corps of Engineers and the National Park and Water Management District scientists who support this work in order to see how well is it going. That interaction, that scientific collaborations there are so tight that it's almost an immediate exchange of assessment to adaptive management. As far as public interaction, that's a greater challenge that the art collaborations have really helped with, drawing the public attention to what is a diatom and why should we care about it. And I think this is a great question that Jan has, because we're trying to understand that. Like it's really important to have public engagement. The public is paying for Everglades restoration a lot of money. It's the most expensive restoration project in the world, and most grandiose. And it's all about supplying more freshwater and freshwater resources to the 9 million people that depend on the Everglades, sustaining this incredible environment for Floridians and the whole world. And so they need to care about it. And they know that alligators and wading birds and these other charismatic fauna are, very visible, and they think about that easily, right? And we've had those are part of the multi-indicator approach that we use for restoration. But what are these diatoms? What is periphyte and why should we care? We're trying to understand that. And that's actually something I think Xavier Cortada is going to talk about next time, is ways of collecting at least qualitative information about what people think when they visit his artwork and why they might then care a little bit more about what's going on in the Everglades. We do try to make the argument to the public when we present this work that we know that you love the wading birds and crocodilians and things out there, but these periphyte communities are what sustain the food web. And through this edibility trophic component of our assessment, we actually are able we are predicting wading bird abundance based on the data that we get. And that's one avenue for saying, you know, the charismatic microflora that we can show you through art or through, you know, some microscopic sessions actually matter to them big megafauna stuff that might be more recognizable.
Right. And Gina has a very practical question about how do you deal with alligators?
Sorry, I've been hogging the answers. I'm sorry, Franco. You take this
No,
one.
not at all. So Gina, as far as alligators go, I think the biggest thing that we remember when we're out in the field is that we have to remember it's their territory and we're just visiting. And so we have had maybe, I think in my 20-some years of working here, maybe 3 instances of close encounters with a gator. I think twice I stepped on one. And the other time, I think it was kind of a one that was more curious than anything, but really decided to test out the helicopter.
Anya, that's Anya, sorry.
Yes,
sorry.
And so when we do get to our field site, we do make sure that we're aware of our surroundings, just as when you're sampling anywhere, if you have to deal with poisonous snakes or anything like that, even poisonous plants. Yeah, but we do keep an eye out for things like that. They have not been an issue. We know when they call and when they're aggressive, what time of the year when they're supposed to be breeding, and we just pretty much just stay out of their way. That's the biggest thing.
I don't think Anya is here. I would love her to tell the one scary encounter where an alligator got up onto the helicopter.
That one, I guess the story behind that one was that the pilot that they had had never seen one. He was new to Everglades work. And so they were pointing out alligators in the water when they were flying over. When they landed and they started sampling and walked away from the chopper, he called out to them and said, "There's a gator near the chopper." So they thought he was joking. The person who was in charge of the throat trapping at that time came around and saw it was a pretty big gator. And so the pilot said, "Okay, well, it's big, then let's get back in the chopper." As soon as they got on and they started lifting off, the gator decided that maybe the float would be well worth a chomp. So it's pretty much just kind of raised itself out of the water just from sheer momentum and almost got part of the float on the chopper. But that was really the closest thing in terms of danger that we had seen out there. I think the bigger thing really is when you throw the trap and you might catch a snake or a catfish when you're mucking around and collecting things. When you're collecting the paraffin, you really have to dig deep under there. And so it's more a matter of like getting bits by a poisonous snake or getting a catfish barb or something like that. But even then, it's we try not to think about it.
Wow! So not only you put your lives on the line for this science.
It's well worth the data,
worth it, isn't it? And so we're already at the top of the hour. I wish we could just keep going because this has just been a fantastic talk to integrate so much of your work. Thanks to both of you again. And if you have a chance, just get on there and look at the chat because people have had some just really appreciative comments to you both. And so thank you to the audience. Thank you for joining us again. And in 2 weeks, We will be, Xavi Cortada will be presenting his work that Evelyn has highlighted here. So we hope to see you all 2 weeks from now. Thanks, everyone.